Hi, fellow foodies. Have you ever heard of the term blue zone? This term was first introduced in a feature story in the National Geographic magazine back in 2005, and it refers to 5 key areas of the world where a large number of centenarians, or folks that live until the age of 100 or older, live. And there were 5 blue zones that were identified. There was Sardinia, found in Italy, Okinawa, Japan, the Seventh-day Adventist of Loma Linda, California, the Nicoya Peninsula of Costa Rica, and Ikaria, Greece. Now, these areas share some commonalities like levels of physical activity, social connectivity, and, importantly, plant-based diets. Now, the types of plants that are included, of course, varies based on the location of each of these blue zones. And in this 23rd episode of Foodie Pharmacology, we're going to take a closer look into a type of citrus that is consumed in Okinawa, Japan, that has some pretty interesting chemistry. Now, obviously, the citrus alone is not responsible for this association with longevity, but it is an interesting element of the diet, and so I wanted to dig in and learn a bit more about it. So, locally, this citrus is known as shikuwasa, and it is used as a flavoring agent in both drinks and food of this region. We're also going to be joined by Dr. Lukasz Ciesla, whose lab actually studies the impact of phytochemical stressors on the body and how they impact resilience and what that may mean for healthy aging. In particular, I'm going to be speaking with Lukasz about an interesting class of molecules that are known as the polymethoxyflavonoids, and these just happen to be extremely abundant in this special citrus fruit that is consumed in Okinawa. The scientific name of shikuwasa is Citrus depressa, and it is a member of the Rutaceae or citrus family. This tart fruit is also known by the names of flat lemon and the harami lemon. The fruit is small and flat, and the tree grows to a height of around 3 to 5 meters. In traditional medicine, the peels or the whole fruits, whether they are mature or immature, are used to treat ailments like indigestion, cough, skin inflammations, muscle pain, and ringworm infections. They are also used to lower blood pressure. The secondary metabolites found in citrus species include flavonoids, alkaloids, limonoids, coumarins, carotenoids, phenolic acids, and essential oils. When you scratch the skin or the rind of an orange or lemon, that very lemony scent is due to components of the essential oil, a major constituent of which is limonene. Now, many of these compounds are important to human health due to their active properties. The polymethoxyflavonoids are especially rich in the fruit that we're discussing today, and there are 3 of particular interest to scientists. These are called nobiletin, tangerintin, and cynacetin. Scientific studies on the compound nobiletin, for example, have shown that it exhibits activity that is linked to improvement of impaired memory, and it has been suggested that this could become a potential leading compound for drug development for the treatment of certain neurodegenerative disorders. In addition to these neuroprotective effects, some of the other pharmacological activities noted for this and other citrus species have included antioxidant, anti-inflammatory, anti-cancer, as well as cardiovascular protective effects. To find out more about the specific health benefits of the polymethoxyflavonoids from citrus, and especially their impact on healthy aging, I met with Dr. Lukasz Ciesla, a natural products chemist and professor. Lukasz obtained his PhD at the Medical University of Lublin in Poland in 2011. After defending his thesis, he worked at the Department of Plant Biochemistry and Crop Quality in the Institute of Soil Science and Plant Cultivation in Poland. He then worked in the Laboratory of Neuroengineering at the Medical University of Lublin. In 2014, he became a laureate of the Foundation for Polish Science program Skills. From there, he went on to work as a visiting fellow at the National Institute on Aging at the U.S. National Institutes of Health in Baltimore, Maryland. In August 2017, he was appointed as an assistant professor in the Department of Biological Sciences at the University of Alabama. His laboratory currently focuses on the study of dietary phytochemicals and their role in healthy aging, as well as on the development of novel techniques speeding up the process of identification of health-promoting compounds that are produced by plants. Lukasz was kind enough to join me by Skype for the show, and I hope you enjoy our discussion. Well, hey, Lukasz, thanks so much for dialing in. It was great to see you this summer at the pharmacognosy meeting, and I'm so pumped to learn more about what you're doing in your lab on neuroprotection and some interesting botanical natural products. So how's it going? How has the semester been so far?
Hi, Cassie. Thank you for having me. And yes, it was. It's a great semester. It's my third year here at the University of Alabama. We just started a lot of new projects. It has been busy because classes start, and I've been teaching a bigger class right now. So it's been busy, but it's great. It's great, and it was actually, it was a great meeting at the and medicine. So I was glad that we had a chance to actually see each other and chat a lot.
Absolutely. Yeah. Well, I was really pumped to hear about some of the work that you're doing with these models to look at like stress responses and how basically how different dietary secondary metabolites from plants can influence resilience factors in the body. And so I thought what would be fun for this episode, well, number one, you recommended a super cool plant of Citrus depressa, this Okinawan sour tart citrus, as the topic for the show today. And I thought it would be fun if we just kind of start with an overview of, in your lab, how do you actually study stressors and how they impact aging and health span? But maybe let's start with what is health span and what is lifespan? What's the difference?
Okay, so first of all, when people look at how we actually age, one of the first factors that usually the scientists look into is actually how much longer I can make my model organism and in the longer run humans to live. So they actually looking how long an organism or a human being can live. So that's what would be a lifespan. But actually, when you think more about it, would you like to live like, well, let's say 100 or 120 years when you are frail, unable to take care of yourself? So a lot more and more people are actually paying more attention to what is called a health span, which can be defined as a period of a disease-free period of your life. So let's say you live up to 100 years, but you will live disease-free. So we no longer.
So it's like living better longer, basically.
Yes. Yes.
Okay, cool.
You just want to, as I said, it's if you ask someone, it's like, okay, do you want to live 100 years? But when you're at your 80s, you're probably gonna get frail. Your mobility gonna decrease. You're gonna have neurodegenerative disease. The quality of your life is gonna decrease. So do you still want to live 100 years? But for 20 years, you're basically going to be dependent on someone. The quality of your life will decrease. So as of right now, the focus is to actually not only make us live longer, but actually healthier when we are actually getting old.
So how do you study that in the lab? Because you can't like have, feeding people in the lab. I mean, I guess you could if you're doing a very long-term study with humans, but that's not really feasible for short-term studies. So what do you work on in the lab, and how do you test these kind of ideas?
So in our lab, we actually work at the very beginning of that whole process when we are actually trying to identify, for example, dietary chemicals that can expand healthy lifespan. So we actually work with pretty simple model organisms. And in my lab, I have specialists who work in Drosophila. So this is a fruit fly. That we can actually easily look at the impact of different stressors on the health span and lifespan of these flies. One of my graduate students actually works in two labs, so she actually works with us, but she also works with the other specialists who actually who are the experts on very simple roundworm organisms as C. elegans. So we actually try to look at how we can improve health span and lifespan in those simple organisms, and then, of course, in the longer run, the compounds that we find to, for example, extend healthy lifespan, they have to be further tested in mammalian models, and of course, at the end, they have to be tested in humans. But as of right now, we work at the very beginning of this whole process, and since we don't really fully understand which, for example, chemicals present in fruit, vegetables, and herbs are good for you, therefore, we are actually testing a lot of different compounds. So you want to have a very simple model that allows you to test a lot of different compounds in a pretty short period of time, and that makes those simple organisms a pretty good model. So that's what we are doing in our lab. We are just testing all those different chemicals using very simple model organisms to see if actually we see any improvement in their lifespan and health span of this model organisms.
Okay, cool. And so I know that one of the classes of compounds that you're working on are these polymethoxyflavonoids that are found in citrus. Um, do these occur in all types of citrus, or is it more restricted towards certain types of citrus, or is that even known yet?
So actually, when we were doing our very first screens to see if we can find any compounds that can be protective against different environmental stressors, we actually noticed that a certain group of chemicals produced by plants actually stood out in all those different assays that we were doing. And actually, one of the first compounds that we have noticed that actually stood out were actually polymethoxyflavonoids. We have like two or three in our preliminary screens, and we saw that, okay, they definitely have a very great biological activity. So let's try to expand on other structures, related structures. And while doing that, we realized that there's something that all these compounds have in common that make them really good protectants from different environmental stressors, at least in all the different models that we have been using. Interestingly, polymethoxyflavonoids are actually derivatives of compounds that are pretty common in all plants. So all plants actually synthesize polyphenols. So this is a group of molecules that are synthesized by all known plants on the planet. However, only certain plants actually acquired the ability to further modify the structures of these basic polyphenols and actually make them polymethoxyflavonoids. So, for example, it's known that plants from the Rutaceae family, like, for example, citruses, have this ability to modify those simple flavonoids into polymethoxyflavonoids. Certain plants also from the mint family, like, for example, sage, rosemary, or mint, actually also have the ability to synthesize those polymethoxyflavonoids. And interestingly, now we then realize if you actually look into ethnobotanical literature, you will see that actually certain plants that actually contain those molecules have been reported to be good for your memory. I remember actually one of your episodes when you were discussing rosemary. Rosemary is for remembrance, right?
Yeah.
We started to think about it like, okay, it looks like there might be some sort of a correlation between the content of the group of these compounds and the possibility of using these particular plants for some neurological disorders. So those polymethoxyflavonoids, actually, to the best of my knowledge, are only synthesized by two plant families, by plants from two plant families. There's Rutaceae, and the mint families, so Lamiaceae. So
That's fascinating because, I'm thinking about the blue zones too, where you have these large concentrations of centenarians, of people that are living to 100 years or older. And, just thinking about those two plant families, two of the blue zones are in the Mediterranean, so very rich in a variety of different Lamiaceae species, those mints, those rosemary and other wild and cultivated mints. And for this particular citrus species of Citrus depressa, it's found in Okinawa, where it grows and people consume this all the time as a flavoring and also in as a food. And that's interesting to me. I mean, you can't, necessarily link causation to that, but it is interesting that you have this enriched diet of, plant ingredients that do have a number of polymethoxyflavonoids in their makeup.
This is actually extremely interesting for us as well, because definitely, and you know it well, that there's a ton of epidemiologic data that clearly show that a diet rich in vegetables, fruit, and herbs is actually associated with healthier aging. And honestly, that if you think about it, we just poorly understand why is that. We actually really do not fully understand what chemicals actually produced by plants are good for us. Definitely, there are a lot that might be harmful, but we actually do not know that which of the chemicals that are produced by plants are good for us and why they are good for us. So, what kind of mechanisms they activate in humans that actually make them good for us. So, for many, many years, it has been promulgated in the field that the major reason that eating vegetables or fruit is good for you because they are rich in antioxidants. And we do know that those compounds actually work as direct antioxidants in vitro. But if you would like to actually get the very same free radical scavenging activity in vivo, we now know that it's actually physically impossible to get a concentration high enough in your cells to be able to actually physically scavenge all these free radicals.
Yeah. So, what this brings up one idea of, so we know these are antioxidants, right? And they serve that purpose for the plant's protection as well. But what about all these different anti-inflammatory pathways and cascades that a lot of these compounds are acting on? We know some compounds are also contributing to gut barrier protection, and now emerging science is showing a link between gut health and mental health and neuroprotection. I think that, like you said, we're just now scraping the very surface of all the different potential ways these could be working. And then to make things more complicated, we're not looking like in a pharmaceutical study at one compound and one potential target. Instead, you've got many compounds in these plants hitting possibly many different targets. What's your favorite target to look at in the lab?
So actually, I'm glad that you've mentioned, because you started with actually innate immunity pathways. And actually what we have observed that one of the major pathways, which is called NF-kappa B pathway, that we have observed to be involved in the protective effects of certain of these molecules. So what actually we're looking at, really evolutionary conserved pathways. So pathways that evolutionary appeared pretty early and they are conserved through the evolution. So all the different animals, simple animals like, for example, fruit fly or C. elegans have it, and then they are also present in humans. And what is actually interesting, we see that, at least with all the different experiments that we have run so far, that these molecules actually act through some of those really evolutionary conserved pathways, and one of them is innate immunity pathway. So this is something that was previously hypothesized. So we think that actually certain polyphenols might have evolved as early as the common ancestor of plants and animals. So when the plants still have the ability to synthesize those different polyphenols. Unfortunately, animals, during the evolution, the genes that are responsible for synthesis of these molecules were edited out. However, we still do have different receptors to respond to them. So
that's fascinating. Yeah.
So, for example, like this is something interesting. Like, for example, if you look at what happens to a stressed plant, it actually, if you look at the polyphenol profile, the synthesis of certain polyphenols actually increases. And this is what those polyphenols, by interacting with different pathways in plants, actually allow the plant to survive under stressful conditions. So now we actually are asking a question, what if the same thing actually happens when an animal is eating those molecules? Are these molecules are also activating or modulating all those different pathways in animals, and also allow them to survive under stressful conditions or enhance their survival under stressful conditions.
So in other words, that's like basically the definition of resilience is, are they better preparing the organism to stand up to some sort of assault or some sort of stressor better than it would otherwise.
Yeah, so this is and the other thing that you've mentioned, and I also loved it because this is something that is actually a hot topic right now, is actually the gut and brain axis. So interestingly, there was a very interesting paper very recently that I read maybe a week or 2 weeks ago that showed that gut microbiota were actually metabolizing a certain polyphenol, and I think it was from grapefruit, and actually modifying its structure. And this modified structure actually was responsible for activating some pathways in cells. And actually, the pathway that they activated was actually focused on the way the mitochondria actually handled in the cell. So one of the things I started to think is like if you look at gut microbiota and then at mitochondria, were once actually bacteria. What if those signals are the way two sisters talk to each other? The sisters, one sister being in a guts, the other one being in, the mitochondria. But definitely there was the paper actually discussed this polyphenol being transformed by gut microbiota and then working as a form of a signal and sending the information to actually mitochondria and enhancing the survival of the cells through the activation of that pathway.
Yeah, that's really a good point. I mean, if you think about the products that we eat, the things that we put into our body, right? You may, as a natural products chemist, you can study those plants in the lab, extract the compounds that are found in the plant, and we can get a good idea on the identity of many of those compounds. But once it enters into the body, there's no guarantee that that actual compound that we see before it goes in the body is going to be the same, because microbes in our gut are all heavy at work modifying these, or they're breaking them apart. And there are many examples of natural products where you can have a glycoside unit or a sugar unit attached to the core compound that bacteria can easily cleave off, and that will change the biological or pharmacological activity of those compounds. The microbial crosstalk, the way they're modifying the things that we're basically feeding them, it's amazingly complex, right? And I think we'll take many years to wrap our heads around all of these detailed pathways that are underway. But I think that is a key part of what's happening in the gut and how our food does so clearly influence our health. It's not just the food; it's also what those microbes are doing.
If you think about it right now, isn't it amazing that this is only right now that we start to realize how important those bacteria are? So before it's just, the way that we were just focusing basically on our cells. But if you look at the numbers, we're actually more bacteria than actually humans. Big avatars for those bacteria. So.
Yeah, I mean, and the science is still emerging. I was talking about this in class today. We were just on the very early, very bare surface of discussing, the role of the human microbiome in our health. And one of the students asked, well, what is a good microbiome? And I think that's a question we can't really answer yet, because your microbiome changes throughout your lifetime. It changes with your diet. It changes as you age. It changes based on so many different factors. And so the way that these phytochemicals, these compounds from plants that are entering into our diet, are actually transformed by the microbiome I would hypothesize are also changing depending on whatever that composition is at the time of contact.
We did not evolve to eat one particular diet. If you look at humans, and we were able to thrive in different types of environments with different types of diets. So is there one optimal microbiome, or there are multiple microbiomes that, depending on what kind of diet you're eating, it's a lot of different factors that can be taken into consideration. And there's one more thing that you actually mentioned about the glycosides, for example. So what we have actually observed is that when we are talking about those chemicals that can signal, that are synthesized in response to different stressful conditions and allow plants, for example, to survive. So usually plants keep them in their cells in a form of a glycoside. So while during the stress, actually the glycoside residues are cleaved and the signaling molecule is actually released and allows to bind to those different targets in the cell and enhance the survival. So the same thing might be actually in humans. So for example, the bacteria cleaving the glycoside residue and releasing those chemicals. And this is one of the problems that we have right now in our research, is that we know more or less know what we are feeding the flies, but what the question we are asking is like, what kind of molecules are actually making it to the brain and actually are responsible for all those protective effects that we are observing under microscopes?
That's a great question. Yeah, I'm just through my mind right now. I'm thinking, what makes up a fruit fly's gut microbiome? Is that known?
Yeah, so actually, we have a group here at the University of Alabama that works with gut microbiota, and they actually have fruit flies that do not have actually gut microbiota. So you can observe. So they more or less know what kind of bacteria grow in their guts, but we basically know, I would say, as much as we know about human microbiome. So it's just a lot of very scattered information without really understanding what's the role of gut microbiota, in the life of Drosophila.
Yeah. Well, I have another question. So you're feeding the Drosophila, these little fruit flies, and they're itty bitty. They're super tiny, right? And so you're feeding them different compounds from nature, different natural products. And you mentioned that you're looking at what's happening to their neurons. So I'm just imagining how small these neurons must be. Can you walk us through how that kind of experiment would work? I mean, what kind of tools do you need to use to evaluate these changes?
So actually, one of the things that we are looking at as of right now, so there are like several sub projects over this big umbrella that we call the role of dietary phytochemicals. And one of the projects we have is actually looking at the neuroprotective activity of certain dietary phytochemicals. So we were wondering if there is a way of using certain dietary phytochemicals to prevent the development of neurodegenerative changes that actually develop in the brains because of different environmental factors. So it's known for the fact that several environmental factors, like for example pesticides, can induce neurodegenerative changes. And as a matter of fact, we are using a pesticide, which is called paraquat, to actually induce neurodegenerative changes in our flies. So in order to check if certain phytochemicals can prevent that, so before we feed the flies the paraquat, so the compound that would start killing the neurons in fruit flies, we are pre-feeding them with certain phytochemicals. And then we are checking if this pre-feeding procedure saved some of the neurons in Drosophila brains. To assess that, we have a lot of different assays. So paraquat is known to kill dopaminergic neurons. And if the dopaminergic neurons are killed, we can observe, first of all, the flies actually, the lifespan actually decreases. So they actually start dying way faster. Their movement, their mobility is impacted. So you can assess the mobility of Drosophila. But so we also look at some biochemical markers. So reactive oxygen and nitrogen species in the brain. But one of the important things that we are looking at is the ability of the phytochemicals to save those dopaminergic neurons. To do that, you have to isolate Drosophila brain. So
I'm assuming you need a microscope and some very tiny tools. Is that right?
and a very patient scientist. So it has to be an intact brain. So as of right now, I have a research associate who is a Drosophila specialist in the lab, and she works with a cohort of students. And as of right now, it's like we have a lot, some of the students that I work with. So we have a certain group of undergraduate students in my lab. So most of them are actually on the so-called pre-med track. So I'm telling them that you're actually, by isolating the brace, you're also practicing a very important skills of patience. And second of all, if you want to be a surgeon, you want to
They'll
be
be
very,
great at
very precise.
microsurgery. That's right. That's great. So, have you observed then, Lucas, with these studies? So you're pre-feeding these flies, then you're stressing them out like crazy by adding this toxin paraquat, and you're isolating their teeny tiny brains. And is it actually helping the presence of these compounds? And I mean, how big is this difference between the control groups and the treatment groups?
Oh wow! So actually, there's a group. So actually, the group, one of the group that we are studying, so the polymethoxyflavonoids. So if you look at the brains of flies that were pretreated with certain phytochemicals and that were not were pretreated with just a sucrose, you can actually see a major difference. So the paminergic neurons are actually organized in certain clusters and Drosophila brain, and you can observe actually that certain clusters are actually totally being safe. So you see the neurons that are normally killed by paraquat. If the flies were pretreated by some of those phytochemicals, you see that those neurons are still there. That would normally be gone if you would just treat the fly with paraquat. So this is so we are actually trying to understand, first of all, well, which actually chemicals are these that are actually responsible for that, and if they're making it through to the actually brain, or is it something that actually might be, maybe these are the metabolites that are actually responsible for this neuroprotective activity? And second of all, what mechanisms are actually involved in that neuroprotection? So what pathways, what cellular pathways are being activated or inhibited by those phytochemicals that allow those neurons to survive, although they are actually treated with this neurotoxic paraquat? And this is actually an interesting question. So one of the things is that we are actually constantly being exposed. I mean, humans and other organisms are actually constantly being exposed to different types of environmental toxins. And the question is there if are we actually less vulnerable to develop all those different neurodegenerative changes if we actually ingest those phytochemicals that we know that can prevent neurons from
Yeah.
degeneration?
So I
So
guess in this model, then you're telling us you're really using an acute type of damage, right? You're giving them a single dose of something really nasty. But as you said, I
yes.
think what humans tend to be exposed to over time from environment and also even behavioral choices and the kind of things that we expose ourselves to, um, we have more of a chronic. Most people have more of this chronic exposure to toxins that could put stress on the system. And so how would you go about do you think that there are ways to study how this could be protective for more chronic assaults or chronic stressors?
So as of right now, we actually that's actually part of the further study. So as of right now, we want to actually understand what are the mechanisms that are involved in the protective role of these phytochemicals. Then I think that that would require special modifying the
Mm.
dosages of this compound that is actually responsible for that insult. So instead of acute exposure, you would probably have to use smaller dosages of this neurotoxic compound and see, first of all, whether it actually causes neurodegeneration. And if it does, how, if we can check if the exposure to pre-exposure to those phytochemicals actually prevent those neurodegenerative changes. But however, here it's actually another interesting thing is that sometimes, and this is something that we actually also observe with natural products, is that sometimes actually compounds that you would normally consider as toxic, if used at the proper low dosage, they actually, you can use it for your own benefit. And this is the phenomenon which is known as hormesis. So something that normally would be very, very toxic at a higher dose, if you use it at a lower dose, and you can actually, in your cells, you can actually induce the response of what we call a stress response cellular signaling pathways that actually activate to protect cells from different type of insults. And we actually, this is something what we are actually also working on, because we have observed proven there are certain classes of phytochemicals that normally are considered to be toxic. However, if you use them properly, and by using properly, we have noticed that intermittent treatment with those chemicals can also provide some sort of a protection, because it actually, by intermittently treating those, for example, flies with those chemicals, we are seeing that the cells are actually, basically, we can simply say, building their
So
resilience. But
this is
this is
different though than the polymethoxyflavonoids, or are you referring to those compounds?
So these are actually a little bit. So we have actually. This is something that we are still trying to figure out, because it's something super new that we have observed. Is that and we still don't know what is the difference, because we have certain polymethoxyflavonoids that actually could be identified as a toxic, but they would require, in order to be classified as a toxic, you have to have certain groups of present on the molecule. So if you have a polymethoxyflavonoid, you can have those methoxy group differently located on the molecule. And depending on the localization, actually, we have observed that they might have those toxic effects or not have them at all. So we are actually at the point of the project that we are actually trying to figure out which of those compounds can be friends. We call them actually friends, and which can be foes. And by actually knowing which group they belong to, we actually can modulate the feeding protocol for the beneficial effects in the organism. Because, as I said, both groups can be used to your benefit. However, you need to know what type of feeding protocol to use in order to help the
Yeah.
model
So
organism. So
there's a lot of research still to be done, it sounds like. I mean, I think that the epidemiological findings so far on citrus and these polymethoxyflavonoids is certainly very compelling, as are some of the early data that you've been seeing with these flies and with neuroprotection. I guess one last question is, we've talked a bit about some of your other studies moving forward. I'm wondering, out of all of this, what's been the most surprising kind of element to your research? Or have there been any aha moments that really, took you, that really popped out at you?
So actually, one of the things that was actually the most surprising was that this huge. So sometimes we've been seeing, a very slight difference in the structure of the molecule, and having had super high impact on the pharmacological activity of the molecule. So we actually have seen that a slight modification of a molecule, if a plant evolved the ability to slightly modify the structure, it can actually turn the molecule that would we normally consider as a friend into a foe, and actually that determines the biological activity, which actually makes a lot of sense, because that's what I've been telling my students. If you look at plants, we sometimes look at plants that are kind of inferior to us. But I'm telling students, like, they are actually dealing with the environment that we also have to deal with in a totally different way. We have evolved, consciousness. We are bipedal. We can communicate easily. So we have a great way. We think, we have a great imagination. But the plants, the way they actually deal with the environment is through chemistry. They are beautiful. They are just amazing alchemists that actually synthesize a plethora of different molecules. And some of those molecules evolved to help plants to survive. Some of them evolved to communicate with other organisms, including mammals. Some of them evolved to actually deter and kill. And sometimes by introducing very slight modifications in a molecule that normally evolved to be a friend, actually a molecule can become something that a plant can use to actually deter potential herbivores and omnivores. So one of the most surprising things that we have seen is just this huge difference in biological activity that we have been observing, even in case of compounds that sometimes look very similar. However, they have a very, very different biological activity. And now we are trying to answer those questions, like, why is that a slight modification changes this biological activity so significantly? What are the mechanisms that are involved, and how we can use this knowledge to actually for our
Yeah, that's
benefit?
great. That's great. And I mean, it's exciting that something that's so accessible to us in our diets, oranges, this specific orange found in Okinawa I don't believe it's as widespread globally yet, but we do know that polymethoxyflavonoids are found in a number of different citrus species. So I think that members of the audience can all benefit from, including some citrus in their diet. And, I think it's exciting to see how the research will evolve, and hopefully we'll get down to some real insights into how this promotes resilience and neural health. Thanks so much, Lukasz, for coming on the show. I mean, this has been fantastic and really exciting. I look forward to seeing how your research comes out. Next time on Foodie Pharmacology, I'll be recording on location from Scotland. I've got some special episodes queued up as I explore the Scottish cuisine from the perspective of the new, the old, and even the wild. I'll be traveling from Edinburgh to deliver a talk at the Royal Botanic Gardens, Edinburgh, and then up into the Orkney Islands to see the northernmost whiskey distillery, and then finally back into the highlands around Aviemore to explore some of the amazing forests and lakes and streams that they have in that region. So stay tuned as more comes your way. I'm Dr. Cassandra Quave, and you've been listening to Foodie Pharmacology, the podcast for the food curious, recorded in Atlanta, Georgia at the Emory University Center for Digital Scholarship. You can subscribe to the show on SoundCloud or iTunes. You can also check out our website at etnobotanica.us/podcast, or you can find me on social media at QuaveEthnobot. Thanks for tuning in. I'll see you next time.